Welcome to the 2025 Bioinformatics Toolkit Overview, where we explore strategic integration and analytical advances that drive modern research. Over the next sections, I'll walk you through optimized workflows, sequencing technologies, cloud solutions, and AI-driven analytics, highlighting how these components fit together to accelerate discovery and improve reproducibility across projects and institutions. This slide outlines a common bottleneck in sequencing: inconsistent lysis and low RNA integrity, which caps downstream accuracy regardless of software. The visuals show a clear workflow from standard lysis to low-quality input, then a solution path using tailored bead homogenizers and extraction kits optimized per tissue. The result is higher yield, improved RIN scores, and libraries with maximal molecular diversity and minimal bias. Here, we compare sequencing platforms and hybrid strategies, showing how short and long-read technologies complement each other. NovaSeq X Plus excels at high-throughput RNA and SNP work. MGI DNBSEQ T20 cuts, costs per gigabase. Oxford Prometheon delivers ultra-long reads for structural variants, and PacBio Revio provides hi-fi accuracy for de novo assembly, together enabling routine hybrid approaches for comprehensive genomic resolution. This slide lays out a pragmatic sequencing strategy and cloud scaling plan: short reads for bulk expression and variant frequency, long reads for repetitive regions, and structural variant discovery, then a modest pilot cohort to align methods to project goals, followed by pay-as-you-go cloud compute for cost-effective scaling. The flow emphasizes piloting before scaling to match resources with genomic complexity. Our core analytical toolkit centers on Galaxy for GUI-driven, reproducible workflows, and R with Bioconductor for statistical power and multi-omics analysis. Galaxy makes teaching and reproducibility accessible with visual workflow builders and no-code interfaces, while R offers thousands of specialized packages for RNA-seq, methylation, and publication-ready plots within a single scripted environment for deep, integrative analyses. This slide presents cloud and workflow solutions that scale genomics pipelines. On the left, an AWS-based NGS cloud provides pre-configured, secure pipelines and shared storage that facilitate team collaboration. On the right, Nextflow enables portable, containerized workflows that run locally or on HPC and cloud, guaranteeing reproducibility and built-in parallelization, so identical results can be reproduced across environments. Velocido uses spliced and unspliced transcript counts from single-cell data to infer RNA velocity and predict future cell states. The central diagram shows velocity vectors and trajectories in UMAP space, revealing differentiation paths. Integration with ScanP and downstream Loom files lets you embed velocity into clustering and pseudo-time analyses, giving dynamic biology insights beyond static expression snapshots. CellBender addresses ambient RNA contamination in droplet scRNA-seq by using deep probabilistic models to separate true cellular signal from background noise. The diagram walks through contaminated raw data, model correction, and recovered cellular profiles. Applying CellBender improves cell calling accuracy, enhances cluster separation, and plugs directly into existing Seurat and ScanP pre-processing workflows for seamless adoption. For clinical and scalable cancer research, this slide contrasts CLC Genomics Workbench and DNA Nexus. CLC offers an intuitive, point-and-click interface and FDA-cited variant reports ideal for hospital labs and regulated workflows, while DNA Nexus provides HIPAA-compliant cloud scaling, secure data management, and advanced interpretation dashboards that support hundreds of cancer samples daily and streamline clinical pipelines. VIA is an integrated variant interpretation engine that merges optical genome mapping, next-generation sequencing, and microarray data for automated variant classification. The visual highlights automated calls for structural variants, copy number changes, and SNVs against curated guidelines, feeding a knowledge database that generates customizable reports and historical case retention to accelerate hematological malignancy sign-off and diagnostic throughput. This slide highlights deep learning variant callers that outperform traditional tools in accuracy and adaptive learning. Benchmark visuals show higher indel and SNP accuracy and reduced false positives versus GATK, while the pipeline flow emphasizes continuous consortium-driven retraining and real-time adaptive pipelines. The net effect is stronger decision support, higher confidence variant detection, and better targeted precision medicine strategies. The final slide outlines next-generation bioinformatics platforms that combine multi-omics integration, containerized workflows, and AI-driven discovery. Containerized pipelines harmonize transcriptomic, epigenomic, proteomic, and imaging data into systems-level signatures, while FAIR metadata and cross-study AI mining shorten biomarker discovery cycles and enable diagnostics that point toward holistic, systems-level biological signatures for translational impact.